Greetings, do you know when did biotechnology started? In 1999, a Hungarian engineer named Karl Erke invented the word biotechnology. But is this the start of biotechnology? The answer is no. As time passed by, different scientists interpret biotechnology differently. But for now, let us move on with the general definition of biotechnology, which is the technological application of biological materials. Biotechnology aims to improve certain sectors such as agriculture, medical and pharmaceutical, industrial, and the environmental. Specific examples include producing drought-resistant crops, recombinant proteins, vaccines, as well as genetically engineered bacteria to treat polluted sites. After World War II, scientists all over the world made groundbreaking discoveries and biotechnology bloomed. In 1953, Watson and Crick published the double helix DNA model. In 1955, the first protein insulin was sequenced by Sanger. In 1961, Jacob and Monod proposed the operon concept, and in 1975, the first monoclonal antibodies was produced by Kohler and Mousning. In 1977, Sanger's sequencing method was developed by Sanger. In 1985, Caramellis developed the famous polymerase chain reaction, which is PCR, and in 1996, Wilmot successfully cloned the first animal, Dolly the sheep. So, what are the main problems caused by the lack of bioinformatics? Certain data could not be published on paper, such as genomic structure and the 3D structure of proteins. If the data obtained from the Human Genome Project was to be put into books, the data would be 200,000 pages long, and this would take at least 26 years to finish reading that data. This means extracting data would be extremely inefficient. The huge amounts of data would need to be stored in databases to be vital for future references in biological research. Anyways, if this amount of data were to be stored in hard copy, the world might simply not have enough papers at all. Nucleic acids and protein sequences by itself have no meaning. It must be analyzed and compared against existing libraries to predict the structure and function. Another problem is that without bioinformatics, the results would not be shared across the world, and this would impede discoveries. What are the choices of bioinformatics tools used to solve stated problems? Multiple tools are needed to allow the databases to function effectively. These tools would allow proper interpretation and analysis of the data.
The database should be searchable, accessible, structured, constantly updated. Furthermore, able to cross-reference, user-friendly, accurate, and precise.
Examples of databases for genetic sequences are GenBank, EMBL, and DDBJ. For protein sequences are SwissProt, ProCite, and PIR-PSD. Examples of databases for others are ECEnzyme, PDB, OMIM, Map9, and Rebase. Examples of tools for sequence alignment is BLAST, ClusterW, ClusterOmega. Examples of tools for genome visualization, CGView Server and GenomeVX. Examples of tools for sequence converters are SeqRed, ReadSeq, and SeqVerter.
Microbiome application in multiple industries. Microbes are very tenacious and can be found in almost everywhere. This could mean different microbes have different proteins that allow them to survive in different conditions. In 1994, US Department of Energy sponsored the Microbiome Genome Project. This showed that microbes have potential for unique applications. Human industrial activities has always been a problem to the environment. It is believed that certain microbes can help to reduce pollution or even better, solve pollution. Bioinformatics applied in bioremediation. The study of genome, which is genomics, and protein, which is proteomics, can be an important key to apply microbes in bioremediation. By using genomics, the specific gene that can be used in bioremediation can be screened in any bacteria. Proteomics can be used to identify the protein function and structure to determine the effectiveness of the specific protein in bioremediation. From this, we can know whether the bacteria can be used in bioremediation and the effectiveness without even performing any trial and error experiments. So how does this relate to bioinformatics? Well, genomics and proteomics relies heavily on bioinformatics databases for gene or protein sequences. Bioinformatic tools are also vital in making sense of the sequences. In a nutshell, effective application of microbes on bioremediation requires bioinformatics.
Outcome of the application. Identifying subspecies is now possible because if you were to differentiate two different strains of bacteria from the same species manually, it is nearly impossible as most bacteria from the same species look alike. Manipulation of DNA at desired condition. Because if the gene of interest is found in a different species of bacteria which might not survive the harsh condition, we can isolate the gene of interest and insert it into the microbes that can survive the harsh condition to be expressed. Classification of phylogeny made easier because different species genome can be sequenced, and by using bioinformatics tool, the similarity can be identified between species to identify their ancestry correlation. So, what is the benefit of using bioinformatics? Bioinformatics can facilitate the process of bioremediation by allowing rapid identification of the types of bacteria to be used in any given scenario. Reduces the cost of bioremediation because there is no need to perform trial and error experiment to screen a useful microbe for bioremediation. Lastly, increase the revenue for industrial waste cleanup company because microbes require a low cost to maintain, yet bioinformatics database and tools are mostly free to use. That's all from us. Thank you for your attention.